Hello, my name is Matthew Garnett. I'm a group leader at the Wellcome Sanger Institute. I'd like to thank the organizers for giving me this opportunity to present today. These are my disclosures. Success rates in oncology drug development are low, typically thought to be less than 10%. And the number of new drugs to new targets is actually declining. And for most genes, it's actually quite unclear whether they could make good therapeutic targets and in what contexts. And so approaches that help to identify and prioritize candidate drug targets could expand the range of targets and improve success rates. And it's worth saying that even a small increase in drug development success rates could have a considerable impact. And so today, I'd like to tell you about our recent work using CRISPR-Cas9 knockout screens that have led to the identification of Werner helicase as a candidate therapeutic target in microsatellite unstable cancers. To identify new drug targets, we performed genome-wide CRISPR-Cas9 knockout screens in 324 cancer cell lines. These include a range of different tumor types, including colon, ovary, breast, and pancreas. These tumor types were selected because they include some of the most common types of human cancers, based on unmet clinical need, and of course, for practical reasons, the availability of a large number of cell lines for our screens. And the cell lines have all been extensively genomically characterized through things like DNA sequencing and RNA sequencing. And using this approach, we're able to identify hundreds of fitness genes per cell line. And in fact, on average, we identify approximately 1,400 fitness genes per cell line. And when you look across all 324 cell lines, we identified 7,470 unique genes that are required for the fitness of at least one cell line. And when I say cellular fitness, I mean genes that either lead to cell death or inhibit proliferation. So the challenge becomes, when you have so many genes, selecting which of these fitness genes might actually make a good therapeutic target. And so to address this, we've developed a very systematic approach to nominate and prioritize new oncology targets. And it was very important for us to be very unbiased as possible in this approach to find the widest possible range of targets. So we began by identifying genes which were required for the fitness of a subset of cancer cell lines, what we call context-specific fitness genes. Now, it's worth noting that genes that are required for the fitness of virtually all cell lines, what we call core fitness genes, were excluded because we felt that these were much more likely to be associated with non-selective toxicity of normal healthy cells. So taking the subset of context-specific genes, we're able to derive a fitness score from our CRISPR-Cas9 screens. And then we integrate these fitness genes with additional orthogonal evidence. For example, is there evidence that that target gene is altered in human tumors or aberrantly expressed? We also looked for pathway level support that a gene is required for the fitness of cells. And finally, we identified genetic features or biomarkers that were predictive of differential dependency on a particular gene in our cancer cell line collection. And we did this analysis when considering each cancer type individually, but also considering them all together in what we call a pan-cancer analysis. And finally, we also integrated tractability evidence or druggability evidence to determine the potential druggability of each target for small molecule or antibody-based inhibition. And so bringing this all together, this gives us a ranked list of targets based on our priority score for each cancer type and pan-cancer. And altogether, we identified 628 priority targets. Not going to go into a lot of detail about all of them today, but we're particularly interested in what we call group 2 targets. So these are targets for which they have a high priority score, but there's also evidence that they're tractable for drug development. And one of these targets was this protein Werner helicase, or WRN, which will be the focus of the remainder of my presentation. So Werner helicase is a strong selective dependency in microsatellite unstable cancers from our analysis in the setting of colorectal cancer cells, in the setting of ovarian cancer cells, or when we look pan-cancer. So each circle here represents the fitness effect of knocking out Werner helicase in a cancer cell line. And we've broken it down into cells that are MSI, so microsatellite unstable, or microsatellite stable. It's worth noting the effect size of Werner knockout in the microsatellite unstable cells is actually very similar to what's been observed for these human core essential genes. To further verify this, we performed colony formation assays from cell lines from 4 different tissue lineages, comparing microsatellite unstable with microsatellite stable cell lines. We're able to show, again, using this very sensitive orthogonal method with 4 independent guide RNAs to Werner helicase, that we have a very profound effect by knocking out Werner helicase in MSI cell lines that's similar to what we see with our positive control, which is knocking out polo-like kinase. And this was true across all of the MSI lines we used in this assay. In contrast, when we did the same experiment in the setting of microsatellite stable cell lines, we didn't see any effect from Werner knockout. And finally, just to say as well that we've effectively confirmed this Werner dependency in MSI cells using siRNA knockdown. So Werner syndrome helicase is a very interesting protein. It has this poorly understood role in maintaining genome stability and is involved in DNA replication and repair as well as telomere maintenance. It's one of 5 RECQ family members, and it contains 2 catalytic domains, an exonuclease domain and a helicase domain. And Werner syndrome is known as adult progeria. So this is a rare autosomal recessive disorder characterized obviously by loss of Werner function that leads to premature aging and cancer predisposition. And so I think what this shows here in these set of images is that people who suffer from this syndrome tend to age normally during childhood until young adulthood, and then they undergo this kind of accelerated aging phenotype and have an average lifespan of 54 years. Microsatellite instability is caused by defects in the mismatch repair machinery. And this leads to the accumulation of these characteristic microsatellite repeats, as well as a very high mutational burden in cells. So the mismatch repair DNA repair machinery is mediated by a cascade of proteins which identify mismatches which are introduced during DNA replication. It recognizes these, it leads to excision of the base mismatch, then resynthesis and ligation and correction of the mismatched base. Now, microsatellite instability occurs across many different cancer types and occurs at a relatively high frequency in some of these. So for example, in the setting of endometrial cancer, stomach cancer, colon cancer, you can see that about 20% of tumors are microsatellite unstable. It also occurs at a much lower frequency across many additional cancer types. So to really robustly evaluate microsatellite instability as a biomarker, we've gone on to expand our analysis of microsatellite unstable models in the setting of colorectal cancer, compiling a new cohort of MSI cell models. We've then used siRNA knockdown and confirmed that the majority of MSI colorectal cancer models are dependent on Werner helicase to a similar extent as our positive control PLK1. You can see that across all of these different MSI colorectal cancer models. Interestingly, though, we did identify 5 MSI cell lines that were relatively independent of Werner helicase. We also went on just to confirm this in our colony formation assay, showing that indeed these 5 cell lines are insensitive to Werner knockdown. And so using these new data, together with our original screening data and data from others, we can build a landscape of Werner dependency across 62 MSI colorectal cancer models. To the best of our knowledge, this is the largest set of MSI colorectal cancer models assembled. We find that 92% of MSI tumors are dependent on Werner helicase, as shown here in all the red models. Now, we've shown that this appears to be independent of whether the tumor is a primary or a metastatic lesion, or derived from a primary or metastatic lesion. It's independent of the mutational status. We've confirmed this in 2D cell lines as well as in organoids and PDX-derived models. And in addition to that, Werner dependency appears to be independent of the driver mutation present within that colorectal cancer. And so collectively, these data really strongly support Werner helicase as a target in MSI colorectal cancers. Now, in our large-scale screen, I showed you that Werner dependency was very prominent in the setting of colon and ovarian cancer, but we can actually also see this dependency quite clearly in the setting of gastric cancer cell lines, and to somewhat lesser extent in endometrial cell lines. And so these are tissue lineages where MSI is quite common typically. We also, though, had cell lines in the screen from cancer types where MSI is much more rarely observed and has much lower clinical frequency. And in this setting, we didn't observe any real strong Werner dependency, suggesting that tissue context may in fact modify Werner dependency. And it's mostly in these MSI predominant lineages where we really see this strong dependency on Werner helicase. So to determine whether loss of Werner induces apoptosis, we compared induction of apoptosis in MSI versus MSL cells using an IncuCyte live cell imager and measuring induction of caspase cleavage. This is shown here for PLK1 in an MSI line and in 2 independent Werner knockouts. We could quantify this effect shown here. This is the induction of apoptosis in the same cell line, KM12. But in some cell lines, we see a very pronounced effect. For example, in the setting of SW48, we see very, very pronounced induction of apoptosis. In contrast, when we look at MSS cell lines, we see very little or no induction of apoptosis when we knock out Werner helicase. To determine whether the loss of a fitness effect was truly selective to Werner and identify potential strategies for drug targeting, we performed functional rescue experiments. So in these studies, we're using either a wild type or hypomorphic alleles of Werner helicase from mouse, which are resistant to the CRISPR knockout. And we had 4 different alleles. We had the wild type Werner gene. We had a version where the exonuclease domain had been ablated by a point mutation, and we had 2 helicase domain mutations as well. And using this functional rescue experiment, we can see that when we knock out Werner, we see this effect on viability. We're able to successfully rescue this effect with the wild type allele, as well as with the exonuclease deficient version of Werner. But we were unable to rescue the phenotype with the helicase deficient versions of Werner. And thus, these point to the fact that the helicase activity of Werner is required for its function, and is an important domain that could be used for therapeutically targeting Werner. We've also examined the effect of Werner knockout on tumor cell growth in vivo using mouse xenograft models. And in this setting, we've used an inducible Werner knockout system in an MSI cell line. So what we can show using the xenograft models is we get formation of tumors. And once we have an established tumor, if we induce Werner knockout by giving the mice doxycycline, we see a suppression of that tumor growth compared to the control arm of the experiment. Using immunohistochemistry of the tumors, we're also able to show that we're able to suppress proliferation of tumor cells as measured by Ki-67 staining. And we also see an induction of apoptosis measured by caspase cleavage. And so these data provide in vivo support for Werner as a target in MSI colon cells. Now, strikingly, using again this Werner inducible knockout system, we see that when you knock out Werner, this leads to massive chromosomal damage compared to control cells. So here is the control cell prior to doxycycline induction, and this is 72 hours later. And basically what you see is massive fragmentation of the genome that becomes almost shattered in some cells. Of course, this is consistent with a role for Werner in the maintenance of genome stability in MSI cells. Now, to investigate the mechanism of Werner dependency, we've also examined the impact of the acute modulation of mismatch repair. And really what we're trying to do is either to suppress mismatch repair or rescue mismatch repair to see if this impacts on Werner dependency in cells. So we used, in the first approach, a set of isogenic cell lines. So it's a mismatch repair deficient cell line where mismatch repair is reconstituted by the reintroduction of MLH1, MSH3, or both proteins together by actually adding back the whole chromosome. So as in our control arm, you can see that when we knock out Werner with guide RNAs, we see this profound effect on viability. And when we add back MLH1, MSH3, or both together, we actually still see this effect. So adding back or reconstituting mismatch repair is not sufficient to overcome the Werner dependency. We've also done the opposite experiment where we take an MSS cell line, we knock out MLH1 with a CRISPR, and then we grow it for several months. And even in this setting, we didn't observe an induction of Werner dependency. And so these data suggest that acute modulation of mismatch repair, at least through these knock-ins and knockouts, does not impact on Werner dependency. And so bringing all this together, our results support Werner as a target candidate, synthetic lethal target, as a candidate synthetic lethal target in MSI cancer cells. And specifically, the loss of Werner leads to growth arrest and apoptosis, as well as this very profound DNA damage phenotype. And we've shown this both in vivo and in vitro. Now, the mechanism underpinning Werner dependency remains unclear, and we're currently exploring two hypotheses. The first is what we call the second hit hypothesis. And this is because we know that acute modulation of mismatch repair is insufficient to modulate Werner dependency. So one possibility is that you need a second hit or an additional molecular change in mismatch repair deficient cells to actually confer Werner dependency. Our identification of rare MSI cell lines, which are resistant to Werner knockout, might help us identify these additional molecular events. Alternatively, it could be due to what we call genetic scars, whereby mismatch repair deficiency over many years generates alterations or structures on the DNA that are specifically require Werner activity to be successfully resolved. As I mentioned, we're exploring both of these hypotheses. Our finding that MSI status confers a synthetic lethal dependency on Werner helicase has now been supported by multiple independent studies. And these collectively have led to drug discovery programs in industry to develop Werner helicase as a target in MSI cancers. So I'd just like to close by thanking the people involved in this group. It's very much a team effort and some fantastic collaborators at GSK, EBI, and the Candiolo Cancer Institute. I'd also like to thank my funders who made this work possible, and I'd like to thank you for your attention. Thank you very much.